Another nuance in cancer drug development is resistance, or that leads to relapse. So relapse being your cancer comes back. So in some patients, unfortunately, you're clear of cancer, but 10, 20 years down the line, they come back. And that's a whole area, again, of research of why cancer comes back. Understanding the mechanisms of resistance of cancer is, again, another module that we can talk about for hours and months. It's just too much. Here we have 4 resistant mechanism, and we'll talk to you about that earlier. But I'll just draw you to the graph here on the left. So ideally, this is disease progression over time, so cancer progression over time. And ideally, what you want for a treatment to be is to slowly kill the tumor over time, long-term benefit, and it will never come back. But in reality, when you treat cells, you kind of inhibit, you kill some cells, may natitira, and so that one will kind of grow back again, and then you hit them with another drug, and then may natitira pa rin, and so it's like you have this cycle of just treating, retreating, and this is kind of very common in cancer, and there's a lot of research as well on understanding the mechanisms of resistance here and developing drugs for frontline, first line, second line, third line, fourth line. I don't know. I don't think they've stopped at fifth line treatment. But yeah, this is unfortunately the sad truth, and this is where, again, research needs to be understanding the mechanisms of resistance for cancer. And so today, again, I cannot talk about everything. We'll touch upon 2 different resistance mechanisms. One is alternative growth signaling, or bypass signaling, and the other one is mutations or altered expression of the drug target. Yeah. So first is this alternative bypass growth signaling. So again, this is the complex version that I will try and simplify. So here we are again. We meet the cancer cell in red. Here's the receptor, yung antenna. Let's go back to the HER2, yung breast cancer receptor that we were talking about earlier. So we know that Herceptin, the drug that was developed from 1984 up till 2000, the 20-year development drug Herceptin, has been developed to inhibit or target the HER2 receptor. So the HER2 receptor, we know, signals downstream. So these 2 proteins, which is called the RAS MAPK protein, this is the pathway. They will signal to the nucleus and instruct the nucleus to proliferate and grow, divide, and all that. So great, that's like, we found a cure for cancer. Herceptin will kill all the breast cancer cells, but no. Unfortunately, there are other downstream pathways. So you have this pathway called PI3K AKT pathway as well, which is another growth pathway in cancer. And so kahit you dampen this response, this will be overactivated, and so that leads to resistance as well. And then breast cancer cells is not just the HER2 receptor. They also have other receptors. They have what we call the estrogen receptor, and that also feeds into the AKT pathway. And so again, this will, kahit ini-inhibit mo to, you also have this receptor that you have to target, because this will also promote cell proliferation and growth. So that's one aspect. So you have the alternative growth pathway. You have other receptors present in the cell as well. And then you also have, the integrin receptors we spoke about earlier, the one that like helps you move around with the feet and the migratory phenotype of the cells. And so these integrin receptors feed into another pathway. They signal to a protein called FAC, my favorite protein during my PhD. And then FAC then feeds into AKT again. AKT will then cause cell proliferation, growth, and all that. So, kahit meron kang isang drug dito, there's other. It's so complex. The cancer cell is very clever. Matigas ang ulo. So they will, find ways to evade, to escape this drug here. And so ideally, we need to combine therapies. And so what you can do is combine drugs that target here, PI3 kinase, drugs that target AKT, combine Herceptin with an estrogen receptor. And this is being done in the clinic, combining Herceptin with tamoxifen that targets the estrogen receptor. And then you can target, again, other proteins called FAC. So this is what I was studying during my PhD, this drug from Pfizer. So combining Herceptin with FAC. So, this is the way forward. You're not just using one drug, you're doing a combination of drugs. But again, the downside of that is side effects. So, if you have to really optimize and carefully combine the drugs. And this is why you have to do the clinical trials, because you can't just combine it. A doctor can't just say, oh, I thought of something. I'll combine this. But no, it has to go to the clinical trial process, because the toxicity of combining multiple drugs for a patient, it can kill as well. And so this is an example of how we tackle, or how the research that we do in addressing drug resistance. So I was talking about my favorite protein called FAP and HER2, and this is just showing you, what we can do in the lab just to show that combination regimens is the way forward. So this is an MTS assay. So you remember earlier yung cell proliferation assay. So I'm using here the MTS assay. And here are, again, MBA361. These are breast cancer cells that have the HER2 in their surface. And so here in black is the Herceptin alone, Herceptin lang. And we treat cells with increasing doses of the drug from 0, the control, to 10 micromolar Herceptin alone. What happens if you combine it with a FAC inhibitor? So you're combining this with this inhibitor, is you actually enhance the response of Herceptin. So here, from here to here, from here to here, from here to here. So this just shows that, FAC is actually a mechanism of resistance to Herceptin, because when you combine, or if you inhibit both of them, you actually kill more cells. And so that's just one way of studying drug resistance. And then another resistance mechanism is, as I said, the mutation or altered expression of a drug target. And so going back here again, complicated, but I'll simplify it. This is the tumor, the cancer cell. So now we move from breast to this is a lung cancer cell, for example. And so in a lung cancer cell, they often express not the HER2, but the EGFR receptor. So again, same thing. Antenna receives messages from the extracellular, the environment, to signal the cells to grow, the cancer cells to grow. So there is a drug. It's called erlotinib from Roche. So this will inhibit, very similar to the Herceptin, inhibits a certain part of the EGFR receptor, and great, like very effective. This was approved early 90s, I think, or maybe late 90s, the erlotinib. But what happens is when they become resistant. So the most common resistance mutation, resistance mechanism is when they have a mutation. So they acquire this, what we call this T790M mutation in the receptor. And what does that mutation do? They will actually alter the shape of the protein in a way na hindi na makakabind yung erlotinib effectively sa receptor. So basically, the drug will no longer work because you have this mutation. So that's one resistance mechanism. And so EGFR here, this schematic on the right, is a receptor that is very prone to mutations. So here they have this tyrosine kinase domain that is like the hotspot for mutations. So, I was just talking about one, the T790M, but there are other mutations involved in in drug resistance. And so now I think in the last 10 years alone, research has led to the development of second gen and third gen drugs. So one of them is osimertinib that specifically targets that mutation. But now I can see in research that there's not just this, there's definitely more mutations. So EGFR receptor is it's so makulit, and they matigas ang ulo, and they will just keep acquiring mutations and resistance. So definitely more research needs to be done with EGFR and lung cancer as well, because they just evade and they just continue to mutate and be resistant and to cancer.